get that?
Yeah.
Maybe people forgot where the room is. Just curious, how does this, so far, already one of the,
Oh, we did something similar to this. I remember we calculated the load units because they really screwed with doing it. So I remember we calculated.
did go
No, we didn't go into the yellow.
Just curious. Okay. I've been using these formulas to calculate P from your data. And Q. I just want to point out something, that this is just simple algebra. If we add these two equations together, you get right? Because they're both equal to 1, so you can put them equal to each other. Okay, so if you understand algebra, this may sort of help you out a little bit. Okay? All right, so where we left off. So looking at how much diversity is there out in the real world. And I pointed out that gel electrophoresis, as far as biochemical variation, is about 30%. That means that usually most organisms, that 70% of the loci, everyone has the same thing. Okay? And there's other types of variation. Something here I want to point out is geographical variation. Some species that have real wide ranges, that they're subject to different physical forces that you go from, for example, southern part of their range to the north. Or if it's up a mountain range, mountainside, those are living at lower elevations compared to higher elevations. They are subjected to different temperatures and a lot of other physical features, and therefore they sometimes have variation that follows some sort of environmental gradient. Okay, and this is called a climb. So when it's along an environmental gradient, there's changes in the morphology of a particular species from population to population to population. That is called climb. Okay, so give an example here of one. In many mammals, like in North America, for example, coyotes have a real large range. And those that are in the southern part of their range, say in southern Texas or even down into Mexico, tend to be somewhat smaller than the ones that are up in Canada. And so there's this gradient of an increase in size from southern parts to northern parts, okay? And it's not just on coyotes, there's a lot of animals that show that type of climb. Why do you think animals tend to get larger the further north you go?
To preserve body heat.
Yeah, it's conservation of body heat, because it gets colder up there most of the time. And by increasing in size, this is a problem of surface area from which you lose heat versus volume. But they don't increase at the same rate. Volume increases as a cube of linear dimension, whereas surface area is a square. And therefore, you have a more favorable, retention of heat if you're bigger. But that, it's a trade-off though. What is problematic about being big compared to being small? You have to eat more food, okay? And so, do you think the resources for eating food are better in the north, where there are short seasons of growing, or in the south, when it's long? Okay, so you see it's a trade-off. I want to point out here that a lot of evolution is exactly that. We never evolve the best of all worlds, because there's multiple forces that are sometimes contrary to each other. And so it turns out to be a trade-off. All right, so that's one. And another client I'd like to point out, this was a study that was actually done by some professors at Stanford University here in California. And so they were looking at stature in a plant called yarrow. The yarrow's all over the place. However, okay, so here's the Central Valley. We're way over here, all right? And as you go up the foothills of Sierra Nevada, up, and they collected plants from each of these elevations. And as you go up, they get shorter and shorter. And then once you reach the crest and go down into the Great Basin, on the Nevada side, they increase in stature as well. Now, the problem with this is, could this be environmentally induced? Or is it genetically induced? I mean, it is a climb. I mean, we do see along an altitude gradient a change in variation. So it is a climb. However, is it due to environment or is it due to genetics? How would you go about experimentally determining that? What would you do?
Get two seeds from the same plant and plant them in different locations.
Do what?
Get two seeds from the same plant and plant them in different locations.
I still didn't.
Get like same plants and there's offspring, put this offspring to different locations.
Okay, so he said to just mix them up, gather from seeds from each of these and move them somewhere else. There's an easier way and more direct. I mean, that would be one approach, but it'd mean a lot of driving around, which I don't mind. I like to drive.
You could just all plant them in the same controlled environment.
Exactly. So that's what they did. They gathered the seeds from these, took them down to Stanford in the greenhouse, and grew all of them under the exact same conditions. Same temperature, same water, same soil type, giving them the same amount of sunlight. And what would that predict if this was geographic or environmentally induced versus genetically inherited? What would you expect the outcome to look like?
Environmentally induced means in the control, all of them would grow the same.
Yeah, the average height of all of them from every place, if it was environmentally induced, they would be pretty close to the same. No significance. But if it is genetically determined, then yes, you would have, they would still grow this way. And that's exactly what they found. The ones that were gathered from up here, high elevation, they still were short. And those at low elevation still turned out fine. Now, what do you think might be the adaptive advantage, the selective selection for short plants at high elevations?
It takes less oxygen?
Well, but the oxygen content isn't that too much of a constraint there. Something else. Any ideas?
Less energy required.
What?
Less biomass means less energy required. Which is like
Well, they get the same amount of sunlight. Okay, they still get.
Is it like wind? Maybe like.
Maybe, but that's not critical.
Water?
Okay, usually high up you get more water. Okay, that would favor growth. Okay, something else. How much time do they have to grow to reach sexual reproductive age? It gets cold early up in the high mountains. Okay, lowlands, stays warm for some time. So there's shorter growing season, okay? Shorter growing season, higher elevation. It starts snowing up there long before it starts snowing down below. And so it's shorter growing season. Therefore, they have to, if they grew to that stature, it would take too long before the cold sets in, and their reproduction would be curtailed. And therefore, they become sexually mature when they only grow just a little bit, okay? And then they produce flowers. So they have to grow fast and reproduce. If they took that long to grow real tall, then they would never make it, okay? So there was a selection against being tall at higher elevations because of the shorter growing season, okay? All right, so natural selection works on variation. So how is variation generated? Now, some of this would be a review from 101A. For one thing, just in sexually reproducing organisms, there are many mechanisms for shuffling genes, shuffling alleles. Both parents are contributing different genetic components to make a new combination. Furthermore, and in fact, some organisms, not animals or plants, but some like chlorophytes, these are the critters you're going to learn about a little bit later on. They may single-celled organisms. They frequently will reproduce only by mitosis throughout most of the growing season. But when the environment becomes stressful, then they will engage in sexual reproduction and meiosis. For the simple reason that by mitosis, how much genetic variation is produced by mitotic cell division? 0. Okay, the offspring are identical genetically. And if everyone growing in a pond, say that this is green algae, everyone growing in the pond is exactly genetically alike, and stress, for example, temperatures drop, and that drop in temperature kills one genotype, it's going to kill them all because they're all identical. So they engage in sexual reproduction, meiosis, which shuffles the alleles and creates genetic variety. Okay, some of those variants would survive, some of them might be affected, okay? All right, so sex, real important. And of course, what's involved there is recombination. That happens during sexual reproduction as well. What do I mean by recombination? What happens with recombination? Happens only in meiosis. Okay, describe the process. Crossing over. Crossing over between what? Between homologous chromosomes, non-sister chromatids, homologous chromosomes, and crossing over. Why is that significant? Because it puts together alleles that came from dad on the same structure with alleles that came from mom. And so it's another way to shuffle up genetic material. So that creates variation too. Now what's really important is unequal crossing over. Because what unequal crossing over can lead to is gene duplications. Now why would that happen? When we have synapses, our two homologous chromosomes happen to align. They align by base sequence similarity. Gene for gene that has similar sequence. But all peppered all throughout our genome and most other organisms' genome are transposons. Transposons. I'm sure Dr. Barnaby talked about transposons. No? He says yes. She says no. Okay, transposons. This is sort of repetitive DNA. He may not have called them transposons. There's retrotransposons and there's DNA-only transposons. These are sort of transposable elements. Did he use that word? Does that ring a bell? No. Okay. Well, transposons, only about 5% of our genome, 5% of our genome codes for protein. The rest of it is regulatory DNA, but we have tons and tons of this repetitive ALU elements and transposons. Transposons, especially retrotransposons. And it's hard to tell evolutionarily where these things came from. Have you heard of retroviruses? Retroviruses, okay. Well, see, the base sequence within these retrotransposons is very, very similar to that of retroviruses. So if we have here DNA, and there is a retrotransposon. This could be transcribed, okay, produce RNA, and it codes for part of this when it gets translated. Codes for an enzyme called reverse transcriptase. Have you heard of reverse transcriptase? I'm sure you would bring that up. Okay, now what this is, it is a polymerase that will then, okay, so once we have this translated, reverse transcriptase, it will then recognize that messenger RNA, and then it will transcribe it into double-stranded DNA. Okay, double-stranded DNA. So this is an enzyme that can use RNA as a template and transcribe it into DNA. Yes.
Is that a protein, the one you put after DNA?
Yeah. This here is a protein.
Okay, and so that attaches the RNA with.
Yeah. The information for making this protein is in the transposon. Okay, this is DNA. Okay, this right up here, DNA going to RNA. RNA, ribosome attaches to it. We get the enzyme. The enzyme recognizes the RNA and will reverse transcribe it into double-stranded DNA. Then this DNA will be randomly inserted somewhere else. All they do is copy and paste. It's copy and paste mechanism. All right? So what you have is multiple copies of a particular reverse retrovirus, and we have many different types of retroviruses. Now, part of our genome is dead retroviruses, or transposable elements. They mutated, they don't move, they're not doing anything, they're but taking up space. And we have a lot of them. My point being is that all throughout your genome, you're going to have sections, long sections, where these transposons are. And let's just look at this one situation. Let's reverse this and get rid of this. Wasn't prepared to talk about this, and we'll go ahead and do it. If we have here a gene that is flanked by the same sequence of a retrotransposon, they can, during meiosis, they can misalign, okay? Because as I said, during meiosis, when we have synapsis, the DNA sequences of the non-sister chromatids, they align precisely by sequence similarity. So here we have two sequence similarities. Now this is sort of I got this figure from somewhere, and it's not quite so accurate because it's showing crossing over between two sister chromatids on the same chromosome. That's not what happens. It's non-sister chromatids. Still, it's a good figure. It still gets my point across. So here we have retrotransposon, and we get misalignment. This here really should have aligned with this. That should have aligned there, but they have misaligned. And then when they cross over, then if we have a gene that's in between them, we will end up with one chromatid that's lacking the gene altogether, and another chromosome that has duplicated copies of the gene in tandem. In tandem means right after each other. Now, what can happen when we duplicate genes is that one of them may mutate and become what is called a pseudogene. Pseudogene is a gene that has a similar or very, very similar sequence to a functional gene, but it is non-functional. That's a pseudogene. Did he talk about pseudogenes? Okay, all right. So I'll get you guys caught up to my style, all right? So pseudogenes. Pseudogenes, there's many ways in which pseudogenes can come about, but this is one, okay? Gene duplication. And we can recognize, or another possibility is that both of these two will diverge in function. Mutations will come up. One of them will keep the original function, and the other one will take on a related but different function, okay? For example, on chromosome, what is it? Not sure. Chromosome 15, I think. There's an array of sequences, 3 or 4 of them which are pseudogenes, but 2 of them code for various versions of the beta globin gene. One of them is beta globin, the other is gamma globin. Gamma globin is expressed in a fetus, not in adult, for forming hemoglobin. And so the 2 sequences of that gamma globin and the beta globin gene, very, very similar to each other, but not identical, okay? And so there's no question that it came by gene duplication. So this also creates new genetic variety. It also creates new genes. In fact, there's several other mechanisms, and I don't have time to go into it, by which genes will duplicate. All throughout eukaryotes, we happen to have a family called G protein family. We have hundreds of these different types of genes that code for a type of G protein. And we have many varieties. And so these have been around for a long, long time. Why do we have so many copies of them? They could only arise throughout evolutionary time by gene duplications, okay? We also get chromosomes that get duplicated. In fact, in vertebrates, probably in the ancestor of vertebrates, we have good reason to believe that there was a total genome duplication about 550 million years ago, okay? Anyway, that's where your genes come from. And of course, mutations, okay? A gene duplication is a type of mutation. Because really, when we say mutations, it's any change in the genetic material. And there's many levels of that. Point mutations, here I'm talking about point mutations. And, we have chromosome mutations as well, all right? But this creates new genetic variation upon which natural selection can operate. That's my main point here. All of these mutations, they're random, accidental, random. You never mutate in the direction of adaptation. That never happens. It might be that we have an organism that has been living in a wet environment. The climate changes and it becomes drier and drier. It does not adapt by mutations coming up with a directionality to make it more adapted to the dry environment. What happens is random mutations, and if some of those mutations allow the organism to be fit for a drier environment, then they would be selected for. But the mutations have no directionality, okay? All right, they're rare and random. Okay, so, yeah, point mutations, this is one type of point that are, sometimes they're harmless, a lot of times. Other times they may be detrimental. If it's in that messes up the sequence, amino acid sequence of an important enzyme, it could be fatal. Or other times it might be favorable. It depends. It really depends. on how it changes the protein for which they happen to encode. Also, something that might be slightly detrimental, but not fatal, slightly detrimental, if we change the environment, it may now become favorable, okay? So changing environments can also sometimes change the selective forces that are happening on the particular gene or allele. A good example of this is sickle cell anemia, okay? And I'll come back to sickle cell anemia a little bit later here. But sickle cell anemia, this is a 1 amino acid change in the beta globin. Beta globin is one of the subunits for hemoglobin. Remember that this particular protein, its function is to carry oxygen, okay, throughout your body. That's real important because your blood is warm. What is the solubility of gases in warm liquids? Not very good, okay? So we have the hemoglobin to increase the solubility. It's a carrier protein carrying oxygen throughout your body, right? It consists of 4 subunits, okay, 4 proteins, 2 of them alpha, 2 of them beta. And so this is an amino acid, 1 amino acid substitution at position 6 from the N-terminus of the beta globin gene. And what it leads to is, okay, we have 3 phenotypes. I'll just say let's just have this HBN, that's normal hemoglobin. The heterozygote, superscript S, the line between there, and then HBS. Okay, what this causes, this particular form of the beta globin protein is under oxygen stress, okay? Exercising, walking up the hill, oxygen stress is very frequently, it'll come apart from the hemoglobin and form long chains linked together. Okay, instead of our normal 4 subunit molecule. Now this stretches out the red blood cell, and also in this particular form, that doesn't carry any oxygen. And so these individuals suffer anemia, okay? They can't get enough oxygen. This is okay. We actually have 3 phenotypes here. These individuals are so incapacitated that they rarely make it to adulthood. They have a high fatality rate. This here is a little bit less extreme, the heterozygotes, but still they would suffer under oxygen stress, some of the red blood cells sickling, okay? But if we change the environment and add something else there, which has happened in Africa and places in India, some tropical areas, in areas where there is malaria. Okay, malaria is due to a organism called Plasmodium. And in fact, for humans, there's 4 different species of Plasmodium that are causative agents of malaria. And they are transferred from person to person by female mosquitoes. Mosquitoes suck blood. Okay, male mosquitoes do not suck blood, so only the females. Okay, why? Because they need the protein to produce their eggs. Okay, so the mosquito will pick up a blood supply from an infected person, then carry the organism, and when it injects into a new individual, it inserts saliva down its beak, which contains these organisms. And this is how malaria gets spread. The Plasmodium then digs into the red blood cells and feeds on the red blood cells. Okay? Now, point being here is that in these individuals, they are a little bit resistant to malaria compared to these individuals. They have a higher survival rate in an environment where there is malaria. Okay? So that's why I'm pointing out. They're living in a different environment, a malarial environment. There's favorable selection on this genotype compared to this genotype. But if you take away the malaria, this no longer has an evolutionary advantage. So my whole point that changes in environment may change selective forces that are operating. Okay, and this is also another example, flies and DDT. DDT was an insecticide that was produced somewhere just at the end of the Second World War. And those flies that happen to have the enzymes in the smoothie R to detoxify, they had a disadvantage without DDT because they reproduced slowly compared to other flies. But once we change the environment and give them an environment that's peppered with DDT, then selective forces have changed, and the slower reproducing but resistant to DDT flies happen to be favored, and they would grow. Okay, so those are usually point mutations. Gene duplications, I already talked about that. I already mentioned the globin genes, and that some of them pseudogenes, variety of them. It isn't just only protein encoding genes where we have point mutations that are really real significant. We also have mutations in regulatory sequences. When we look at the distinctions in the proteins between chimpanzees and humans, there is very, very, very little difference. A lot of our hemoglobin, hexokinase enzyme, histones, etc., almost have identically the same amino acid sequence. There's some differences, but it appears that really in some of the major differences that we're starting to find out between chimpanzees and humans have been in regulatory sequences. Regulatory sequences. And one example here is, this here is, it's not a gene, it is a name of a regulatory sequence that is expressed only in limb bud cells in the early embryo. When I say limb bud, they will turn out to be the hand, okay? So there's not too many mutations in vertebrates when we compare this all across vertebrates. The base sequence is about the same, but what we find in humans is about 17 different point mutations. That's really strange. From chimpanzees to horses to whales, there's not that much difference in the sequence. So why do we have so many mutations in the human enhancer? So this is an enhancer. Did Dr. Barnaby tell you anything about what enhancers are? Okay, so it's a control region. And what is the significance of those mutations is that they're expressed in the early formation of the hand. And we believe that those expressions, and then later, once the hand is formed, the gene is shut down. However, humans have a very dexterous hand compared to chimpanzees. We can really manipulate things, and so it's believed, not absolutely certain, but it's believed that those mutations have a lot to do with our dexterous hand, right? Another control region, actually this is the name of the gene, but there is an enhancer, it's called by the same thing, that is expressed also in the early embryo. And in humans, we had a mutation in the enhancer for this gene, so that although the gene is there, it's not expressed in humans. It's been a loss of function mutation. Now, what that loss of function mutation has led to, they've been able to do experiments with this, with neural tissues, is that it increases the number of neurons that have been produced in the brain. And that may have a lot to do with a higher, larger brain that humans have evolved compared to other animals. All right, so control regions can have a lot of significance here, okay? Now, so we get these areas that produce genetic variation, but given natural selection, what is natural selection doing here? In fact, I didn't point this out yesterday, but natural selection, it also has a predictable pattern of how things change. If we have just two alleles, let's go back to this type of graph where I have allele frequency of P and generation. If we start up here, 9, and let's say that it is the recessive allele that is being selected for by natural selection, then you would find over generations a decrease in P, right? Eventually, if the selection is really large, you will lose it completely. All right, so with natural selection operating like that, it's operating on variation where one morph is favored and the other is not. You wouldn't expect us to have too much variation in normal organisms. Variation should be eliminated. But I pointed out yesterday that there's about 30% of our alleles, 30% of our genes are polymorphic. So how can we maintain that high polymorphism in the light of natural selection? Well, there's several mechanisms here that can maintain that value. What is the fact that we're diploid? Okay, so if we're diploid and there's selection against the recessive allele, not the other way around, not against the dominant allele, it is against the recessive allele. Once again, we have A, little a, little a, little a. It is the little a that are being, individuals that are being eliminated, okay? Selection against that. Even, now this frequency will drop, okay? Q will drop. But if we have a big enough population, we still have little a hiding out here in the heterozygotes. This is complete dominance. So these have the dominant phenotype is favored, and therefore even though we're eliminating the little a, we still have a little a that is protected by diploidy, okay? And so the frequency will be real low, but it may be there, okay? All of us have about 10 alleles that are lethal. Not the same ones, but they're recessive and they're in very, very low frequency. Now, if you ever were to have those alleles homozygous, it's fatal in the embryo. The embryo will never make it. Then we have about 6 to 10 of them, each and every one of us. But the only way they would ever become homozygous, because the frequencies are so low, is with something like incest, okay? Brother, sister, or brother, father, or I can't do that, brother, mother, or whatever. Close relatives that would have that rare recessive allele, okay? All right, so that's one way. Another one that's a little bit more common is balancing selection. And I already gave you one example of that with sickle cell anemia. It's the heterozygote in areas where there is malaria that is favored. And what is the heterozygote? Well, it has both. It has both alleles. And so in the homozygous condition, there's selection against the dominant allele. In the heterozygous condition, it's against the other allele. Actually, here we can't talk about this. Balancing selection is not where there's complete dominance. It has to be incomplete dominance. Incomplete dominance, selection against the homozygotes on each side, but favorable selection on the heterozygote, which preserves both alleles. Both alleles stay in the population. All right, so that's balancing selection, and I pointed out that happens here. This is what a normal red blood cell should look like when they sickle due to forming these long polyunit chains. That these do not carry oxygen. And this here is a map showing malaria areas, and the dark purple is the frequency of the sickled allele. We're still keeping it low, 10%, but you see it's only in areas that are coincide with high rates of malaria. Okay, now there's another form of balancing selection that's called frequency dependent. So both of these, heterozygote advantage, we balance, okay? But there's something here that's called frequency dependent selection. then this also leads to balancing. And an example here, this is just one made up, stated that birds are visual predators, many of them, and they will frequently, with their prey, if they focus on one particular prey, they like to be very efficient, is that they will form a search image for what prey it has. So let's say we have a bird that is preying on a certain species of butterfly, and let's say that that butterfly comes in two different morphs, colored morphs. One of them is yellow and the other is white. If yellow is very abundant compared to white, they will form a search image for the yellow one. Okay, focus on the yellow one because they have to catch them real quickly, and white happens to be low. So we now have selection against yellow, and as yellow goes down, and it's the white ones that survive and produce more white offspring, the balance will change. Once yellow becomes rare enough, they will switch their search image and go after the white, okay? So as they keep switching from what is most frequent, attacking what is most frequent, then both forms of the butterfly are maintained, okay? Another example, this would be a real world example, is a type of orchid that is called elder flower orchid. They come in two different color morphs. Most of these are usually found in mountainous areas, so there's a yellow morph and a purple morph. Now, bumblebees and any other pollinator, there's a relationship, symbiotic relationship, mutualistic relationship that has evolved since Cenozoic times up to now between flowering plants and bees and their pollinators, because these are going there for, not for the pollen, but at the base of the flower, there's something called a nectary, which produces nectar. Okay, and the nectar is what they're going for. However, to be successful in pollinating, because the bee will come in trying to get to the nectar, and they bump against the stamens in the flower and pick up pollen. Now with pollen, you're going to be really amazed at plant reproduction later on, because they're really wild in their reproductive strategies. But that pollen contains sperm, okay? And that pollen, then with the bumblebee or any other bee, then goes to another flower of the same species, tries to get the nectar, then some of that pollen will fall off on the carpel, the female part of the flower. Sperm will go to an egg at the base, get fertilization, an embryo, then a seed, okay? The thing is, in order for that to work, the flower should not produce enough nectar to fill the belly of the bee. Then they'll just pick up the nectar, go over and take a nap, all right? And you don't want that to happen. You want it to go from flower to flower, so they give it just a little bit to keep it coming back. Now, orchids are cheaters. They don't produce hardly any nectar. And bumblebees, they still remember things, but they don't remember things for a long, long time. So a bumblebee comes to one of these purple flowered orchids, finds out that it has nothing there to reward it with. It gets pissed and says, I'm not going to go to purple anymore. And then it will go to a yellow flower. And then once it's at the yellow flower, it gets nothing. It gets mad. It says, I'm not going to go to yellow anymore. And then it will go back to the purple. Okay, so it keeps going back and forth like this. All right, so if we have, too bad I don't have purple here, these flowers, let these blue ones be the purple one. And these are the white ones, or the yellow. Okay, so our bee, first of all, visits this, but there's nothing. So it will now go to that color, nothing there. So it'll go back to that color, and then nothing there. It'll go to that color, nothing. Go to there, nothing. To there, nothing. To there, nothing. To here, nothing. You see any sort of pattern here compared to purple versus yellow? How many visits are the yellow ones getting compared to the purple? Blue up there is the purple, green is the yellow. The flowers that are in the lowest abundance are getting visited multiple times. The ones that are more abundant aren't getting too many visits. So this here is sort of a negative frequency dependent selection where the phenotype that is in lower abundance has an advantage, because the more visits by bee, the higher probability of its pollen getting passed on. Okay, so they're favored. So as generation after generation goes on, the yellow flowered dwarf increases, and the purple decreases, and we get a reversal. Okay, so it's balanced. And in fact, in most populations, this they don't fluctuate too much, but it's a form of balancing selection, frequency dependent. Okay, third way in which genetic variation in the light of natural selection, where variation can be maintained, and this is called patchy environment. Most organisms live in ranges where it's not uniform. There's a lot of micro environments, and in these micro environments, the selective forces may differ. Okay, and just as a made-up example here, let's say that we have a lizard that comes in green versus yellow form, and that we have a patchy environment. There's forest, microhabitat, grasslands, and what happens, we'll say this in California because most of the year grasslands are yellow. So the green morph is more conspicuous, less camouflaged than grass, whereas the green form is pretty well camouflaged if it happens to be in the forest, and the other way around. Let's say these things are being eaten by foxes. So if we have a green form that's in the grass, it gets higher rate of attack. But if the green form is in the forest, it's okay compared to the yellow one. The yellow one suffers more attacks in the forest region. And of course, these lizards go back and forth, because the microhabitats are not that large. The whole point here is that it balances out. Okay, you would never get rid of, completely eliminate the green over the yellow, because they have opposite types of selection depending on what microhabitat they happen to be in. And that would help maintain variation. Another one, 4. This is a biggie, because having the assumption that every single trait, every single character is under natural selection is actually false. Most of our characters are variable because they make no difference. They make no difference. And they don't affect survival, they don't affect reproduction. And a good example in humans is fingerprints. No two individuals have exactly, even identical twins don't have the same fingerprint patterns. The ridges on the finger, okay? And they have nothing to do with survival and reproduction. And so it's neutral. Neutral, and therefore free to mutate. Okay? Now, I don't know why I put that up there. Oh yeah, because talking about neutrality. And the beta hemoglobin, I've already talked about it before with sickle, because that's the mutation here in the sickle cell anemia at position 6, amino acid 6. We know of other mutations, substitutions in that same codon, and they don't result in an amino acid, or they do if they result in an amino acid change. It is, the two amino acids are so much alike that it doesn't cause any problem. Okay, and therefore it's neutral. And so we know of 99 different mutant forms. They're not real common, but they're there. They've been found in the beta globin gene in humans, and most of them do not cause any sort of anemia. Okay, they're perfectly all right, neutral. Okay, so just as a summary here, okay, we've already talked about this, but I'll go ahead and just go over it again. So adaptive evolution, only natural selection is the only microevolutionary force that leads to adaptation, a closer fit to the environment. And this term, often hear that, survival of the fittest, it gives the idea of, let's see there, I think there was a poet who said, nature raw in tooth and claw. Okay, anyone know who that poet is? Nature, have you ever heard that phrase? Nature raw in tooth and claw. Sort of mean, isn't it? Okay, it gives you the impression, even the survival of the fittest gives you the impression of everything, a bloody environment, everyone tearing each other apart. That's not really so. What fitness means is reproductive success. And being reproductively successful doesn't necessarily mean that everyone's tearing everyone apart. Sometimes it is that way, however, okay? But barnacles, these are types of crustaceans that they're sessile as adults. They're, put that word up there. Okay, sessile, meaning that they stay in one place. Okay, so, and they don't move. They're filter feeders. And so they attach themselves to rocks or the hulls of ships, etc. And they stay there. They glue themselves and they just take in seawater and filter out of it the eggs and small organisms that happen to be in the marine water. That's what they feed on. For reproduction, therefore, they don't cut each other up. They release their gametes into the water. And so here the competition is between receptors on the sperm and eggs. It's the sperm and eggs that have to compete to form a larva. The larva will swim around, and then eventually it competes with other larvae for living space. Okay? It has to find a substrate on a rock, place where it can fit in. And that's where the struggle happens to be, not tearing each other apart. And furthermore, flowering plants, they compete with each other for pollinators. And so you don't see flowers jumping up and tearing off each other's petals. All right, so it's this is still a struggle of the fittest. All right. Okay, talking about that, what do we mean when we talk, say, fitness? Well, Darwin used this word, and biologists defined it a little bit better afterwards. There's two different forms of fitness. Darwinian fitness, all of these are measured by the genetic contribution to the next generation that phenotype or individual makes. Okay? So Darwinian fitness is the contribution of an individual. So an individual plant that would produce 200 seeds versus another plant of the same species that would only produce 100 seeds. The one that has 200 seeds has greater Darwinian fitness. It made a greater contribution to the next generation. All right, relative fitness, however, when we're talking about phenotypic groups, is more useful, okay, much more useful. Because here it's the contribution, the average contribution of all the individuals of one phenotype within the population compared to the average contribution to the gene pool of the next generation of another phenotype. And this has been mostly what I've been talking about. I haven't been concentrating on that at all. Okay, mostly this. Relative fitness. Okay. All right. Now, all these examples that I've been given, I've assumed that it's a single gene trait. It's a character that is determined by one gene locus. In actuality, there is no such thing as a single gene trait. All genes are sort of in, if it's a protein encoding gene, a lot of times it's contributing to a metabolic pathway in which there's several genes that happen to contribute to the products of that pathway. However, what we're calling a single gene trait is in these particular pathways, there's maybe one particular gene that has an overwhelming effect on the entire pathway. And that makes it look like it's just one gene that controls that character. But then we have a bunch of other characters that really, they're not determined by a single gene, but by many genes. Okay, these are called polygenic. Did Dr. Barnaby say anything about polygenic? Okay, all right. You know, so it's, they're quantitative. It might be in the case, in fact, this is a model. We know that the model isn't quite right for skin color. Skin color is due to the deposition of melanin. It's a pigment that's in fibrocytes that happen to be in the skin. And from all humans, the melanin is melanin is melanin. There's no difference. What's really different is the amount that happens to be deposited. And back in the 60s, they had a model that maybe there was 7 different genes, each of which have 2 alleles, a dominant, recessive, and the amount of melanin was controlled by how many dominant alleles you had at those 7 loci. They were additive, okay? And therefore, individuals who have the highest amount of melanin in the skin would have dominant alleles, homozygous dominant at all of the loci. Those with the lightest skin, homozygous recessive at all 7 loci, okay? Now, light skin does not mean albinism, okay? That's something different. But then we have all these different combinations in between, all right? Now we know that that's not true for skin color now, it's more complicated than that. And that's not quite as additive as what I'm talking about, but we're gonna assume an additive type of environment that is polygenic. So, in fact, most of our characters happen to be quantitative and polygenic. And usually also when it's that type of character, such as skin color, intelligence is also polygenic. It's not just a single gene. Usually these also have a high environmental component, okay? Especially intelligence. Intelligence, yes, there's a genetic basis to it, but the inheritability is not as high. There's a long and strong, very strong environmental influence on intelligence, what we think is intelligence. All right, so usually these types of quantitative characters would follow a bell-shaped curve, a normal distribution, okay? For example, in humans, stature. Let's go with stature. Stature is definitely polygenic. You can start, we have very few short people, some a little bit taller, some a little bit taller, then some real tall, and then short, or no, average, and then not too many tall, okay? So stature is sort of follows a bell-shaped curve if we have a large sample size. Okay, this is average height, this is real tall, this is real short. Okay? And this is how these are distributed. So, but that's several genes for one character. How does natural selection work on polygenic characters? Because up to now I've been talking about single gene traits, where we just have 2 alleles at one gene locus. And now we're looking at many genes that interact together. How does it work? Well, there's 3 different modes of selection that can happen in these types of situations. One is called stabilizing selection, and before discussing this example, which was a true good example, just show figure from your textbook, shows it. Okay, this particular figure is looking at tail length. Okay, tail length, the tail feathers, it's polygenic as well. There's not just one gene involved there. But what stabilizing selection is selection against extremes. So if its tail here is too short, it's a disadvantage. If it's too long, it's a disadvantage. And so what happens is that the mean does not change. The mean of the trait does not change, but it becomes more narrower. Okay, the distribution does. This is stabilizing. And a good real life example, before modern medicine. This is important, before modern medicine, human births were always between 3 and 7 kilograms. Okay, if a baby was born less than 3 kilograms, it did not live. Okay, but today it can. Okay, we have incubators, they're in the hospital, they can exist. If the baby, when the woman's given birth to something larger than 7 kilograms, the baby didn't survive, and usually neither did the mother. Today, we just cut them out. okay, cesarean section, all right? So modern medicine has changed that, but still, this is stabilizing selection, okay? Each end of the extreme is selected against. Another thing can be directional selection. Okay, directional selection, I'll show on the slide, is when one extreme is selected against, say this is being selected against, then what happens over time is the distribution will shift toward the other extreme. Let's again go back here to figure, and this here, directional selection. Selection against one end of the distribution leads to it being shifted towards the other direction. And in this case, the mean of the distribution does change, okay? It shifts toward the larger size. Example of this is during Ice Age in Europe. During Ice Age in Europe, as glaciation increased over several thousand years, then the average size of black bears, they also increased because of the selection against smaller bears because of the colder climate, okay? And then this is rare. It's hard to come up with any example of this, is diversifying selection. Diversifying selection, I can only, I think, yeah, Galapagos finches, on certain Galapagos islands, there's a species of finch that on those islands they have two beak sizes. They're either big or they're short, okay? Let me go back here to show what this is. So diversifying is against the mean, so that the two extremes are the ones that live and survive. Now, with these finches in the Galapagos Islands, they live on a particular island where their food sources, they only have two different foods. They eat seeds. They eat seeds. There's only two different plants that provide the seeds that they can eat. One produces very, very small seeds. The other one produces very large seeds. And so, beak size for those who have the large seeds, they have to have real large, hard beaks to be able to break those seeds. The birds that have small beak sizes, they can't feed on those big seeds. They have to feed on the small seeds. The ones in between that have intermediate beaks, they're sort of left out of the game because their beaks are too big to be able to handle and pick up the small seeds, and their beaks are not tough enough to be able to crack and eat the large seeds. Therefore, they're selected against. And so we have the two extremes. And this leads to a bimodal distribution. Okay? All right, so this is on quantitative, polygenic characters. Okay, so really on exams and quizzes. Now, this is not going to be on Monday's quiz. You know, that ended that yesterday. But on exams, you're going to have all these. What I do a lot of times, I'll do a scenario. Okay, I'll say, give you a bunch of information about a particular population, what's going on there, and I'm wanting you to come up and tell me what mode of example of concept is being displayed here. Okay, so really think about and keep straight these mechanisms that I've been referring to as single gene traits versus polygenic traits. Okay, because some of them, when you study, you'll see some of them have some similarities, but there's also differences. Okay, with selection, I'll do just one last one.
Can I ask a quick question?
Yep.
So I noticed in the graph it says disruptive selection, but.
That's the same thing.
Diversity, so
Yeah,
same thing.
yeah. So, disruptive, so they use the word destructive, okay. Diversifying, disruptive, either word I'll use.
Sure.
Okay, because they're the same. You'll find both of them in the literature. Now, this is a problem with biology is very often you find different people using different terms that they've invented for the very same thing. Yeah, and that will happen sometimes. Okay, and so sexual selection. A lot of animals, not plants, but a lot of animals are sexually dimorphic. And what I mean by sexual dimorphism is that there's some physical feature other than the genitalia, that differs between males and females. Okay? For example, in humans, Males can grow hair on their face, females do not. And so, but this has nothing to do with reproduction, okay? So it's something that's come about by sexual selection. So males and females may differ in size. In mammals, a lot of times the males are bigger than the females, but not 100%. In some other animals, and I've said in mammals, but in some other types of animals, it's the other way around. The females are bigger than the males. And plumage that means feathers. So I'm talking about birds. In birds, such as bluebirds, I find them out here, western bluebird, the females are pretty drab, just sort of brown and gray, and the males are real pretty. They have this bluish wings and orange belly and everything like that. And manes in lions, that's a male. Female doesn't have that type of coloration. Antlers in deer and moose, you find them on males, not females. Females don't have them, okay? So this is sexual dimorphism. Now, why does that come about? Already Charles Darwin, he wrote a book after his Origin of Species that addressed this idea of sexual selection. And it's related to female choice, okay? Females invest a lot in reproduction compared to the male. All the male provides is sperm, that's all. It's the female that has to raise, put in all the energy to produce the offspring, and a lot of times after birth, care for it. And the males can just run away. All right, so the female wants to invest in a male that has good genes. Now, I'm putting this sort of in a teleological way, as if these things are thinking through the problem. That's not the case, okay? It's just that selection, natural selection has led to this, and I'm just interpreting a little bit that way. Nonetheless, the females have to be choosing, okay? And therefore, to figure out what male is going to have good genes that will be that could use to pass on to their offspring, because it's a big investment. They have looked for their indicators of good genetic fitness. So peacock, it's a male, and females don't do this, okay? So just think about a peacock or peahen, okay, the female. Would they want to mate with something like this, or some male peacock with one little crooked black feather? Okay, this is an indicator of probably pretty good genes, right? So it's not that they think of it, it's just that it's selected for it. And so this sometimes the females choosing which males to mate with has pushed males to have all these elaborate devices, okay? Yes? Okay, just a second, let me get a little bit closer. The mask is obscuring things.
I was asking, is there any truth in here? Like, is it actually true that this peacock probably doesn't hurt you?
Usually, yeah. Okay, because if it can provide, because the thing is, are these actual signs of better genetic quality? Yeah. If they could afford to produce this type of stuff, then the rest of the genes in there are probably pretty good as well. Yeah. So they usually are good indicators. And so here, mandrill, okay, that's a male. Females do not have all that beautiful red and blue hair. They didn't dye it either. Okay, that's, yes.
Although in cases like the Irish elk, it can end up being detrimental.
Yeah, it can be, exactly. It can be detrimental. Because Irish elk, it's not even an elk, it's a deer, but it was in Europe, and their antlers became so big that it may have weighed them down, their head down, maybe. Okay, that's been one explanation, but others have come up with something else as well. You know, it's still extreme, and I don't see how they can keep their heads up. All right, lion, mane, that's a female, drab, drab, drab. And this is a male, my, look at all that stuff. And these are males, they compete, okay? And so this is pretty common, okay? So anyone have seen any videos of sage hens? They're not around here. Sage hens have, yeah.
Are they the ones with those like big sacks on their
Yep,
chests?
yep.
Yeah.
And, so they're on the high plains, okay, on the other side of the Rocky Mountains. Actually, even up in Wyoming, they have it. They're on their way to being extinct because of ranching and farming, humans altering their habitat. But they will have these, they'll come together in a big group. Females will be there, and a bunch of males will ruffle their feathers, and they have all this chest that goes way out here and real colorful, and they do these dances and everything like that. So, and it's the males that are doing that. The female will say, "I don't like your steps. Get out of here." And the female is choosing, however, who to mate with. All right? Okay, so just some things here to think about. Said this many, many times, and I did emphasize it. Adaptive evolution, that's natural selection, okay? And the mutations are random, but they're still important, even though I've stated in my 5 conditions of the Hardy-Weinberg formula that as far as changing allele frequencies are not important, but mutations are incredibly important with creating new genetic diversity. And reproductive success is really what is important here. Okay? Yeah, and oh yeah, this last thing, I've been looking at single characters, but really natural selection works on the whole phenotype. And therefore what the result is not the best of all worlds, a Panglossian world view, it's a compromise. Because some natural forces favor one direction of selection, others favor a different direction, and it's a compromise. The whole thing's a compromise. As long as it works, then that's good. Okay, so I can start the next slide here. Any questions? Incidentally, I have office hours right after this, okay? And it can be, you can come online. Let me just explain how to get there. And, or to my office, either one. But when you go to Canvas, did anyone look at your Canvas account? Do you have a link there for my office hours, okay? So just let me show you where you need to go to get in. Should you want to come that way. And for future office hours, it's going to be the same. You can either come in person, or you can come here. But you would go to the link to the office hours, and there is something here called Tech Connect Zoom. That's where you have to go. Tech Connect Zoom, and there will be, I don't know why it's doing that to me. Maybe it's because it's just mine. Yours might be a little bit different. But there will be, right now I just only have Thursdays up here, but I'll put in the others, and this is how you get access. Now, when you go there, you will end up, first of all, in a waiting room, because if I'm with someone else, you may have to wait for me. Otherwise, I'll let you in. I'll be there. Okay, so let me go to the start, at least get a start on the next topic. Okay, speciation. Okay, so I'm not going to dwell on this one because I just said that on the last slide of the previous lecture. Except for here, we've been looking at microevolution. Now I'm going to change and look at macroevolution. Because just like gradualism, a little bit geological gradualism, a little bit of erosion here, or more erosion there, deposition there, over long periods of time can create really, really big changes. Same here with microevolution. Over millions of years, it can create big changes. So we're going to be looking here at macroevolution and emphasizing that it's the environment that selects for characteristics. Now, an overview here of microevolution. Since we're looking at long periods of time, the environment changes, climates change, continents move, and so forth. And therefore, the natural forces that select for certain phenotypes will also change over time. The more stable places to live is in the ocean. Their changes aren't so drastic, but they do happen. But terrestrial habitats were really subject to a lot of environmental climate changes. Okay, now one of the outcomes of microevolution or macroevolution over time is the production of new species. Okay, species will originate, and so this is what I'm going to concentrate on here. Is about where do species come from. So as far as macroevolutionary change, we're looking at lineages. Now, when I say lineage, it is a collection of interbreeding populations that have passing their genes from one generation to the next, and then to the next, and to the next. It's a lineage, okay? All of the populations evolving over time, okay? They're changing, exchanging genes through sexual reproduction. And so two things can happen here of changes, anagenesis and cladogenesis. So anagenesis is just within the same lineage, gradual morphological change, okay? And usually a paleontologist will come out and they make, this is the form that exists now in our yard, but this is a fossil form, okay? It existed millions of years ago, okay? So a paleontologist might find this, and since they're unaware or do not have this information in between, seeing the gradual changes that took place through here, then a lot of times they'll give this a different species name, okay? However, if we had complete information, as far as I'm concerned, because I'm a hardcore cladist, and you'll find out what cladism is a little bit later on, is that this here is really the same species. It's just changed anagenetically, okay? It's the same evolving gene pool, all right? But still, paleontologists usually don't have every little fossil along the way. And therefore, frequently, some fossils in the past, even though it belongs to the same lineage, will often be named a different species. So anagenic change is morphological change, genetic change within the same lineage, without splitting. Cladogenesis still involves anagenesis. It's still change. However, the lineage splits. This is cladogenesis. Splitting of the lineage, divergence. This produces new species. So cladogenesis is equivalent, absolutely equivalent to speciation. Okay? And so we're mostly going to be looking at this process, cladogenesis. All right, and furthermore, how rapidly does speciation, cladogenesis, how fast does it happen? Okay? Darwin, he was a gradualist. He believed that lineages would split and they would gradually diverge from each other. Take millions of years to become morphologically distinct. Back in the 1970s, however, there was a Harvard University paleontologist named Stephen Gould and another one, Niles Eldridge. They were mostly studying marine fossils and the fossil history of marine animals such as trilobites and clams is pretty rich compared to terrestrial animals. Terrestrial animals' fossil record is a lot of times not so good. And what they saw is they would be looking through geological time and having the same type of trilobite comes up to here, and then all of a sudden there was two of them, okay? And then all of a sudden up here, they would stay the same until a certain point, and then all of a sudden there was three of them, okay? They thought that stasis, staying the same, was the rule, and that evolution, speciation, happened in a very rapidly in a very short period of time, okay? And their model was called punctuated equilibrium. Okay, rapid speciation, and over a very short period of time, followed by long periods of stasis. Stasis meaning not a whole lot of change, okay? Now, which one of these two modes, or tempos, not mode, tempo meaning time, of speciation is correct? Actually, both of them are. And you have everything in between. Okay, these are just two opposite extremes of what happens. Some, we believe, some speciation happens very rapidly. Others can be very gradual. And even within the same lineage, it might be gradual over this part of geological time, and then all of a sudden we get rapid speciation. Okay, so both of these can happen. All right, now since we're talking about speciation, this is going to be a biggie. What is a species? Biologists have been arguing about this for about 300 years. And it's very hard, because of the diversity of life, and on Earth, it's very hard to pin down exactly what we mean by a species. In fact, a species in bacteria, we have to define it much differently than we have in sexually reproducing animals. Okay, so there's a lot of different species concepts. For Linnaeus, it was a morphological species. So two things were different species if they had a certain morphological character, and the other one did not. Okay, so it's based on just the morphology. They look different. Okay, and we can point to a particular thing. This here has long canines, and that one over there has shorter canines. Therefore, this is one with long canines, must be a different species. Other than that, they look pretty much the same. Ecological species. A lot of people were unhappy in the 1960s, 1970s, 80s with morphological species. Although that's sort of strange, because when a person working in a museum collection trying to figure out how these organisms that they've collected happen to be related to each other, usually they end up using this to define what the species it happens to be. But ecological species, two different species are ecological species if they occupy different ecological niches. Now, that gets to be very, very abstract. Now, I said that a little bit differently than I had up here. Some populations are the same species, regardless of what they look like, if they occupy the same niche. If they occupy different niches, they are different species. Okay? Now, what is a niche? It's the totality of the living space of an organism. Okay, what it eats, where it lives, how long it lives, what its enemies are, what it feeds on, etc. The totality of its living space. Okay, and that is almost impossible to describe. All right, but nonetheless, it's sometimes useful. Other idea is a phylogenetic species, and this is that all members, all populations that come from a common ancestor, belong to the same species. Okay? This is also sort of hard to put into practice. So all these are not mutually exclusive of each other. Okay? Phylogenetic species could be an ecological species, and also it could be a morphological species. It's just only the criteria by which we're defining, drawing lines between populations to say this is one species, that's another, might be a little bit different. What, especially with animals and with plants, but more so with animals, the species concept that is in almost everybody's textbooks, and what we're going to be concentrating on is what is called the biological species concept. This was established by Ernest Mayr. He was a Harvard ornithologist. So he studied birds, and he also was very big about speciation. He made very major contributions to this ideas of how it happens. And so his biological species concept, and this is quotation directly from his book, species are groups of actually or potentially interbreeding natural populations. So he was really focused on population biology, and that they are reproductively isolated from each other. In other words, reproductive isolation is his key to what is a different species. Okay, two species, if they are reproductively isolated, are different species. If they're not reproductively isolated, they belong to the same species. Now, this gets to be a little bit tricky because when I say reproductive isolated, does not mean that they cannot make and produce offspring. That's not what reproductive isolation means. Okay, and I'll clarify that. Obviously, some species are that type of isolation. Try to cross a mouse with an elephant. It won't happen, okay? Never happen. So they're really isolated. But this becomes a problem about maybe two different species of frog that happen to be very similar to each other. Are they different species or are they just different varieties of the same species? Well, usually this criteria then sometimes gets a little bit difficult to apply. Nonetheless, so the criterion here is reproductive isolation. And reproductive isolation under natural conditions. Natural conditions where they lived in the world, all right? If they're isolated, then we have two gene pools in which alleles are not moving back and forth between the two gene pools. There's no exchange of genes. And as I said, it's not exclusive of the other types of ideas. So what about lions and tigers? Are they different species? In zoos, you can put them together and they produce ligers. A hybrid between a tiger and a lion, so they can reproduce, but under natural conditions, they usually don't. There's something else there in their behavior in a natural environment that keeps them apart. And those are called reproductive isolating barriers. And so on next week, Tuesday, that's what I'll go into, is what are these barriers to reproduction that keep species apart? I'll finish this slide and this is where I'm going to stop. Plants, this biological species concept is a little bit more difficult to apply because plants are developmentally more plastic. What I mean by that is that sometimes you can hybridize not only different species of the same genus, but sometimes different genera of the same family. And this is because it happens with the development, okay, during the embryological stage. Animals are more complex structurally compared to a plant, and therefore, if there's a little bit of misinformation, genetic misinformation in the formation of an animal embryo, it can abort, okay? It just won't go. Plants, however, seem to be able to adjust to those differences, and you get hybridization. And then bacteria, that's a problem, because bacteria don't reproduce sexually at all. So this idea of biological species concept where reproductive isolation is a criterion doesn't work for bacteria at all. Okay. All right, this is where I stop. And I know some of you might want to ask questions, but let's move out of the room because there's another class that's going to come in immediately. And